Okay, it's great to see everybody out here today. Elizabeth is an engineer, and she did chemical engineering at McGill for bachelor's and master's. Then she went to Stanford, did civil environmental engineering, but I don't know if it's, I don't know how many engineers are in the room, but often there's a disconnect in ability for engineers and scientists to communicate. And I think that Elizabeth does an outstanding job of breaking down those barriers. She has both engineers and scientists in her lab. And I think that's something that's really important. It's also something that we've been trying to do within CBB. She is the director of BioZone, which is a very impressive collection of scientists and engineers in chemical engineering and applied chemistry at Toronto. She has a number of honors, including being a member of the Canadian Academy of Engineering and Royal Society of Canada. And this year, she is a Killam Prize winner. I think that we are very fortunate that Elizabeth could fit us into her schedule. I know that she's leaving tomorrow for a trip to Brazil. Thank you very much for being here, really, to kick off this important week. Thank you.
Thank you. Thank you, Trevor. Is the microphone on? Can you guys hear me? Okay. So yeah, thanks very much. I'm really pleased to be here and to share really some of my experiences doing exactly what Trevor just said, which is trying to look at things from an engineering perspective and a scientific perspective at the same time, and finding sort of the sweet spot of how we can get some great scientific ideas out into practice. I don't know what happened to. Let me make sure I got this right. Which is the laser pointer here? There we go. I don't know what happened to the logo. This is supposed to be a circle, but somehow or another it got stretched and messed up when we moved over to this computer.
New age.
So I struggle always when I'm presenting, because you never know who the audience is and what story of the many stories. So I've tried to blend a few stories together today. But since it's for Biotechnology Week, I thought the first thing to do is to Google up biotechnology and figure out what it is. I should know, but I wasn't really sure. And there's a number of reasons why I wasn't sure what biotechnology really means in most people's minds. And I'll tell you one of those stories at the end. But this is a UN definition here. Any technological application that uses biological systems, living organisms, or derivatives thereof to make modified products or processes for a specific use. And I like the second one actually a little bit better because it actually adds in this twist, which is we want to improve our lives and the health of our planet. And I think that we all kind of can resonate with that, I think. But this is fine. You can read those words. But ask yourself, when you hear the word biotechnology, what comes to your mind? What's the first thing that comes to your mind? Money. Okay, that's good. That's good. You can do one of these word map type things here, or you can just look at the first thing that comes up on Google, okay? But really what it revolves around is DNA, and if you look here, it says altered or something like that. The next biggest word here is, I can't read it here, altered, and genetically modified or something like that. And so there is this notion that biotechnology is all about manipulating DNA. And so I'd like to maybe take a little bit of look into that a little bit more. And so this is all stuff you guys know. I mean, biotechnology is ancient, and if you look at review articles on biotechnology, they always classify it in old stuff that we've been doing for zillions of years, and then the new stuff that has all this promise, right? But these old things are pretty darn good. These are really nice things. And so we shouldn't throw these out in favor of the things that we're thinking about in our mind. We need to consider what we learn and what we know about all of these things that have happened. It's been 10,000 years since we've been trying to domesticating animals, and I've got here, even wastewater treatment that is so dear to my heart. This is an old technology now, in the way we practice it, and so it's important because when we talk to the public about biotechnology, the first thing they think about is genetic modifications and all of the specter that frightens people. And this does this whole field a huge disservice. And so we need to work on that collectively to make sure that this part of biotechnology and all the good that we can do is really highlighted. Not that there's no good down here. I'm not saying that. But it's just important that we keep these things in mind and perspective, because this is certainly the images that come up. All right, so so I thought, what would I like to tell you guys about in terms of my romp through the land of biotechnology? And so I thought maybe to start with my first job and take you through a little bit of my encounters with biotechnology. And so, as you heard, I did my undergrad in chemical engineering, and I really did chemical engineering because I had no clue what to do, and it just seemed like something that did everything, so I would just do it. And then when I graduated, it was the first oil crisis, and there were no jobs in traditional petrochemical field. And I happened to land myself a job working at Seagram's making whiskey. And I didn't mean to do this. It was the only job I got, and so I took it. And it actually turned out, in hindsight, fast forward many decades, one of the greatest things that ever could have happened to me. So I worked there from 1985 to 1988. Seagram's had been around a lot longer here, 1857. And I learned a ton of things, and there were some significant perks. And the science behind whiskey making is actually fascinating. I mean, what happens when you sit something in a barrel for 30 years or 20 years? And how do you plan economic? You said about money. Somebody said about money. How do you plan for a market that's going to be 15 years from now when you actually open this keg, right? Or this barrel. So at any rate, what did I learn? So I learned how to mill and cook grains, and fermentation and microbiology and yeast and distillation, which was nice. That was a nice engineering thing. And at large scale, where you have to walk up many flights of this distillation column. But also things like how flammable flour actually is, and ethanol, and how much precautions you have to make when you actually flick a switch in one of these rooms. You can't actually do that. You have to have a safety switch. Economics, we talked about that already. And also, I learned a lot about the value of data, and all of the numbers, and that chemical engineering and many things is really just an accounting of mass and energy. And you're just keeping track of that. And if you don't have good numbers, you can't make good projections. So I learned a lot. This is the manual I was given, 1965, and it had these beautiful diagrams of what was going into the cooker and things like that. And I still have this book. It's really actually a treasure. It smells like ethanol, by the way. And so, ethanol fermentation is just this beautiful place to understand really what fermentation really means. We think of fermentation as meaning you put it in a fermenter. So anything in a fermenter is fermentation, but no. The true definition of fermentation is what happens without oxygen when you essentially get the atoms of the parent molecule reorganizing, part of them getting oxidized, part getting reduced, with no external electron acceptor, as it were. And so that's really the definition of fermentation. And you can see it's just such a beautiful process, catalyzed by little microorganisms, yeast, and bacteria, and and microbes. So how does this large-scale process happen? You grind the corn in a mill, and you cook it with hot water. Now you add enzymes in here. You have to cool it then before you can ferment. You add the yeast. You get the fermentation, but the beer that comes out of here, it's called beer even in a whiskey making process because it's only got about 6 or 7, maybe a little higher percent ethanol. So then you have to distill to get something that can go into a whiskey bottle. And so you have multiple columns, and the huge distillation columns, a big part of the cost, okay? And then there's, of course, this pre-blending, maturing, which is putting in those barrels for different years, post-blending, bottling, and all this. And then at the bottom of the beer still, as it's called, you get what's called stillage, which is all the parts of the grain, the protein, and the husks, and all the stuff that didn't volatilize into this area here, all the solids that are remaining. You concentrate them a little bit and can make this wonderful product here called distilled dried grains, which actually probably is worth more than the ethanol you make. It's a good animal feed. And Then this thin stillage then has to go through, you can recycle some of this water sometimes, or you have to take this condensate and treat it through wastewater treatment processes. So I learned all stages of this process when I was working there. And in fact, one of the requirements when you're working at Seagram's was that you had to give the plant tours, because there was always groups of people, little old ladies, whoever you want, coming to give tours, or students above drinking age. At the time, this was in Montreal, so it was 18 and above, or, well, whatever. And so we'd give tours all the time, and that was a really fun thing to do, because in explaining something to somebody else, you realize what you don't know, and you get better and better at it. So that was my first foray into biotechnology, and I got very interested in wastewater treatment systems because of it. And there was an interesting connection to Waterloo, because Waterloo also had a Seagram's plant, and there's actually Seagram's Avenue not far from here, and the Seagram's Museum is here. And so I was working in Montreal, but the plant at Waterloo had to close down a number of its wells because they were contaminated with trichloroethane, or TCE. And they could no longer use that water, that groundwater, to blend into their whiskey because it was tainted. And so I didn't know that I would later work in that area, but when I was already at Seagram's, I was already encountering this problem. So I got very interested in wastewater treatment and decided that I didn't want to make a career out of making whiskey, and went and did my PhD. And I went, fortunately, I was just lucky enough to get accepted only in one place. I went to Stanford and worked for this remarkable individual whom many of you know, Perry McCarty, who is the father of wastewater treatment, anaerobic digestion, sanitary engineering. He's a civil environmental engineering, and it goes on and on. And this is what he taught me, that civil engineers have saved more lives than all the doctors in history developing clean water and sanitation systems. And so, I think this is a message to remember about process and scale, and I guess how one can have an impact. And obviously, clean water is something that we really need to think about. So another flow diagram, because that's what is important. And this process became very familiar to me through my studies at Stanford, but actually even before that, and subsequently. But here's your influent, right? This is the water that comes in. This is your sewage, toilets, and everything else. A bar screen. So who has never been to a sewage treatment plant? So just put it in your things to do, your bucket list, okay? To go to a sewage treatment plant. It's a fascinating thing. You should know where your sewage is going. So the first thing that happens is old boots and bicycle tires and things like that are screened away, and old rags are screened away. And then you have a grit chamber where sand is removed. And then you have this a primary clarifier which settles some of the organics that are big enough to settle. But the big process here is this aeration tank, which we call secondary treatment. This aeration tank coupled with a clarifier that's a settling tank. So you have this aeration tank. All the dissolved solids are consumed by the microorganisms. You have to add a lot of oxygen, and the solids are settled, or the bacteria grow, the microorganisms grow, and then in the clarifier you settle them, you recycle some of them, and so on. And then from the top of the clarifier comes your clean effluent that you may or may not have to sterilize before, or not sterilize, but disinfect before going into the environment. So this is this process that's been around for more than 100 years, and it really works super well. And here's some pictures in case you've not seen one. This is actually in Bangladesh. I mean, look, these are all the aeration basins. So you can see the size of this kind of sewage treatment plant. And then these aeration basins are just full of fine bubble diffusers in the bottom, and it's a huge amount of energy. So 50% of the cost of running one of these is in the energy for the aeration. And the other big problem is solids handling. So the solids that come out of the bottom of the secondary clarifier here have to be dealt with. You compress them in a belt thickener. And one of the processes that's now widely used is anaerobic digestion. And all that does is decrease the volume of solids by about 50%. Produces a little bit of methane that helps you run the plant. So it has been a huge improvement in just simply dumping the solids. But the solids handling is 90% of the headache for an operator, for the operation of these things. So although this technology has been around for a long time, I think there's a move a move afoot to think, is there a better way to do this? And we'll come back to that idea a little bit. But one of the things I wanted to talk about mostly are these anaerobic processes, like the anaerobic digestion that's there. And this paper, right, is from 1978 or 79. Again, Perry McCarty here, and a number of other authors you may or may not recognize. This very simple test that maybe some of you are still doing, called the serum bottle test. And it's just such a wonderful test that's been around for so long. But you basically put whatever ingredient here, anything you want to digest anaerobically, you seal it really tightly so that it is anaerobic. You may or may not, you may need to inoculate, and you measure how much gas is coming out in a very simple way. And it's still used, and it's still very powerful. And we understand now that in these anaerobic digestion processes, a complex cascade, a food cascade and different microorganisms catalyze these different steps down to these compounds like acetate and hydrogen and CO2 that are the primary substrates for the organisms that actually make methane. And I think maybe you guys know all this already. I'm not sure. I'm never sure which audience I'm speaking to. But I think it's just fascinating that these methanogens that belong to the kingdom Archaea, have really only used these very simple substrates. And so for the carbon that's locked up in these complex compounds to end up as biogas, methane and CO2, they have to go through this process, get broken down first by one set of organisms, and next passed over. There's this handoff. And it's a really beautiful system that was described many, many years ago. This says 1983, but it was actually Perry McCarty was talking about this even before that. And what's interesting is that you see this figure of the stages of decomposition of complex organics. That's the ultimate carbon cycling, right? The ultimate repository for carbon or recycling of carbon. You see these images in, this is from an fermentation process, but you see it in Brock, Biology of Microorganisms. It looks a little bit more developed, so you have these cellulitic and hydrolytic bacteria here, and fermentative bacteria, and homocetogens, and other fatty acid oxidized, they break it up, but then the methanogens at the very bottom, okay? But I like the first thing that they wrote here. This particular picture here was first described in anaerobic digestion years earlier by an environmental engineer, and this is from a microbiology textbook. So, there's this work together, right, between what we're talking about, between understanding the science and understanding the application and the breaking something down into a carbon and energy balance, which is basically what this kind of thing does. And if we take back to our friendly molecule glucose and we figure out what happens in this overall process, it's again very much like the ethanol fermentation. You get a reduction and an oxidation. Part of the molecule gets reduced to methane, part of the molecule gets oxidized to CO2. Although it's brought about by multiple organisms. And so when I was doing my PhD, I was actually co-supervised by a microbiologist. Her name is Dunja Gabriel-Galic, and she introduced me to the notion of groundwater contamination and the challenges with groundwater. And so this was at about the same time, or a bit later actually, than groundwater became a big thing here at Waterloo with the whole groundwater center. And so these late 80s and so on, the groundwater was really starting to become a serious issue, especially in Silicon Valley where I was, but also here. And so there was more and more, there's a lot of interest in that at the time. So now we're looking in the subsurface, and now you have, let's say, something like gasoline, which is a lighter than water hydrocarbon, so it floats on water. And you get these kinds of plumes, and the groundwater flows and entrains the plume. And if you have a well over here, you might pick up some of these gasoline components. And the challenge was, well, what is the fate of these hydrocarbons when they get into the environment? Because there's not a lot of reaeration possibilities, not a lot of oxygen that gets in here. And the big question was, what happens to these hydrocarbons anaerobically? And you can look at hydrocarbon degradation. Okay, so gasoline or oil, hydrocarbon. And if you do the mass balance, okay, let's say you have a spill. So this is it's not that big of a spill. So at a gas station and the tank spills, you might have this much, 2,700 kilograms, whatever the number is, okay? To get enough oxygen to stoichiometrically match this kind of equation, you need to pump in millions of liters of water saturated, because oxygen is not that soluble. It's only 8 milligrams per liter, and it's a huge amount. So the prospects of doing this underground, it's possible to do this in a bioreactor above ground, but the prospects of doing it underground are really challenging. And moreover, you produce a huge mass, more mass of bacteria than you do of the oil that you started with, okay? And so in the context of the subsurface, if you started pumping in oxygen, you're going to plug your aquifer pretty quickly with biomass. Okay, and you do that. I've actually I've done that in a well. When you put in too much new stuff in a well, and if there's too much oxygen, you'll plug the screens in a well. And so, it is a challenge. And the question of the day back then, and it's still a question today, is, well, what is the fate of many chemicals when there's no oxygen? And so, you know, the story started off as saying, well, let's look at these aromatic hydrocarbons, because they are the most water soluble. They're the ones that drive cleanup. They're very toxic. Benzene is probably one of the most toxic components in gasoline that's actually mobile, because it tends to be more soluble than some of the other things. And the good news is that theoretically, anyway, you can convert these to methane and CO2. The free energy of the reaction is negative, meaning that it actually is releasing energy and that the organisms could actually theoretically grow. There isn't a big difference between toluene and benzene, and yet what we observe in the field is a huge difference. We find that toluene can go anaerobically in these kinds of methanogenic systems readily, but for whatever reason, benzene is a lot more difficult to go, and it's not really an energetics problem. So since that time, working with Dunya and Perry, I've put so much stuff in bottles anaerobically, so many dirts from so many different sites from around the world, including Brazil, where I'm going tomorrow. But I think one of the really cool concepts in all of these anaerobic studies, and actually in microbial communities in general, is this concept of syntrophy, which I think is something that needs to be defined carefully. You know, it actually means something very specific. It means to eat together, basically. Syntrophy is kind of a nice thought. But you know, in the case of benzene, we know that the methanogenic methanogens can degrade acetate and hydrogen, but that acetate and hydrogen has to be produced by other microorganisms first. So there's definitely this break here. And if you calculate the free energy under standard conditions, you really, this particular reaction is highly unfavored. Okay? You won't get it to happen. And the only way you can get this reaction, that the organisms that might do this fermentation reaction here, is if the hydrogen or the acetate concentrations are drawn down very low concentrations. And so, for example, here I just put some numbers, but what actually happens in these ecosystems is you get a sharing of the energy. So that minus 60 kilojoules per mole of benzene gets basically split between these three clades of microorganisms. And that's actually what happens. And the hydrogen concentrations and acetate concentrations that lead to this calculation are actually what gets poised in the environment. And so it's a really lovely, and that's really what syntrophy is about, and is when you have this dependence. These methanogens can't get access to acetate and hydrogen unless this happens, so they can't grow. And these fermenters can't grow unless the methanogens are there drawing it back. This kind of example, you can find lots of other ways. It's most well described for this interspecies hydrogen transfer, but I think it's a recurring theme with lots of other metabolites that occur in these communities. And it's one of the reasons why the communities function better as a whole than as any individual. So the reductionist microbiologist tendency to try and work just on this particular methanogen, you know, you lose a lot. And there's almost no way to work on this particular fermenting group, because there's really no better way than using methanogens than to pull these things. If anybody can find a way to really keep a hydrogen very, very low in such a system, that would be a great breakthrough. But the interactions are numerous between these groups. And you can see it, this is then the sort of overall kind of process, but if you look at a micrograph, the rods that are blunt like this are methanocyeta, and you can see that the organisms attach with a variety of extracellular materials, and who knows what's being exchanged there. They're in close proximity. And so when I finished my PhD working on gasoline, I went to work with a graduate from microbiology here, Dr. Dave Major, who got his PhD in microbiology here, and he did working in consulting. And I went to work with him in Guelph, and so came back to Canada. And they didn't care about gasoline one bit. It wasn't driving anything. By the time I was working on, got into consulting, the notion that hydrocarbons could degrade quite naturally and that natural attenuation was really a great process for dealing, at least with gasoline spills and things like that, was really quite strong already. And the EPA had put out some guidelines on to how long a plume would go and things like that. And so there was less interest from, it was less push to clean up that, but there really was this issue of trichloroethene. And so these were the kinds of solvents, these were the chemicals, and they still are actually the most commonly encountered organic pollutants in industrialized areas. I mean, hydrocarbons are very common too, but these tend to have a higher toxicity associated with lower drinking water limits that you have to get to, and also are really difficult to clean up, primarily because they're more dense than water. And so when you have a spill, they don't stop at the water table. They move down and down and down, and your lateral extent of contamination, and you get a very deep, difficult to access, difficult to map, and even know how do you clean up something you don't even know where it is. And it's just a really challenge. And they're really, I would argue, there are very few really good ways of cleaning up sites contaminated with TCE, and very few sites that have actually been properly cleaned up. So back when I started this work, we actually went to a site that's just down the road from here and put dirt in bottles. And for this, we wanted to understand what was happening to TCE. So this is a long time ago now, and it's before anybody really knew how TCE could dechlorinate. And we knew, though, it would most likely happen under anaerobic conditions. And it wasn't, I don't know if you can see the line here, it looks a bit washed out, but TCE went down, and you get these intermediates, so trichloroethene, and you get the dichloroethene, vinyl chloride, and ethene. So you get a dechlorination of the ethene backbone. And it became clear over the years that this was a reductive process, and it was that you needed to provide an electron donor. At the time, we provided methanol just because it dissolved TCE really well. And so we called this culture KB1, and I was working with Dave Major still, even I'd worked in consulting, but then went back to academia, and we worked on a field demonstration to show that actually you could stimulate this kind of dechlorination in the field, but moreover, that these dechlorinating organisms were not ubiquitous, and that actually adding in these kinds of microorganisms that you'd grown up in a reactor first might actually help. So there was this bioaugmentation demonstration done at an Air Force base in Texas, where we injected culture into a well and tried to do this mass balance, try to be sure that we could account for how much we injected, how much grew, how much PCE was dechlorinated. Try to do a control. We had a control plot as well that didn't have. So doing an experiment, a proper experiment in the field is always a challenge, but this particular experiment worked relatively well, and we were able to extract DNA from the monitoring wells and show that you actually could only account for the loss of dechlorination by growth of microorganisms, and we modeled the process and got a nice demonstration out of it. And on the basis of that, Dave Major and others decided to form a company, and they called it Serum. This was back in 2001. They've had a rebranding, so now they have a new logo, so that they've been around long enough to have that happen. And the culture is just, the company is still going strong in Guelph. And they market a whole range of cultures for inoculation. And again, these are just enrichment cultures from groundwater samples that we just coax to degrade TCE or other chlorinated compounds. And so they've developed techniques just based on quantitative PCR to track the organisms when they inject. They've developed ways to monitor the activity in their laboratories and ways to inject it into the ground at a contaminated site, where you bring it in a beer keg, basically like this, a tank of argon, and this is a wellhead, this black thing behind, like this is a well here too, and you just pressurize one end of the tank and push the culture down into the groundwater. And so I should say that the Air Force Base demonstration was a contaminated site about this underground, about this 2 or 3 times the size of this room, and we inoculated with 15 liters of culture to give you kind of a scale, an idea. Okay, so now they grow up lots and lots of culture in larger vessels, 100 liters vessels, and many, many of them. And I think at some sites they've injected hundreds and maybe even thousands of liters. And so the technology has taken off to some extent, as much as clients need help in remediation. And Serum is not the only company that sells culture. There's quite a number of others that sell these kinds of enrichment cultures. And so Serum itself has been in these countries and probably more than 400 different sites at this point in time. So it's really fun, and we continue to work together, troubleshooting and optimizing and figuring out why it works sometimes and sometimes it doesn't. But in all cases, if you mix the microorganism and the contaminant and the electron donor together, you get dechlorination. But the challenge in the field is mixing. It's not the biology. It's the same problem with any technology. It's the mixing part of it. So on the science side, at the same time, there's been a lot learned about this process, which we now call organohalide respiration, because it's now understood that why this dechlorination, this beautiful, and this particularly this last step, which really turns something chlorinated into something non-chlorinated, and very, very much and non-toxic. Ethene is absolutely non-toxic. The microorganisms that do this use these compounds as electron acceptors, just like we use oxygen. So they breathe, so it's called halo respiration or organohalide respiration. And there's all kinds of genera that do this. And now, of course, once we start to know this, you can start trying to understand what's happening in a consortium. So it's exactly the same pictures I've shown before. These organisms that dechlorinate actually use hydrogen. They compete with the methanogens. They use hydrogen as their electron donor and TCE as their electron acceptor. And they just fit right into the same picture of anaerobic decomposition that we talked about before. And assuredly, I should say that these kinds of organisms, these dechlorinating organisms, have been doing this since the dawn of time on all the naturally occurring halogenated compounds that are out there. There's a number of things we've learned over the years in research about dechlorination. We're looking at it to try and speed this reaction up and clean up more sites, but that the critical cofactor for the enzyme is a coronoid, and the coronoid is not produced by the organism itself, so they rely on the community members to produce this coronoid. We also know that there's not just one dihelicocoides, there's actually a geobacter, which also dechlorinates, but actually we've also found out that there's multiple populations of dihelicocoides, many different ones within the enrichment culture, so it's not one. Many genomes have been sequenced. The first one was in 2005, and it was a very reduced genome, 1.5 megabases roughly, a huge number of reductive dehalogenases. I don't know why those are little V's there. I'm not sure what's going on there, but and also hydrogenases. And so this is just a snapshot of a number of organisms that are organohalide respiring, that are capable of this reductive dechlorination for energy. They grow. The nice thing about it, I should have mentioned, is that they grow on at the expense of this reaction. So when you inoculate that 15 liters we put in, it turned into a much higher population because they grow on the chlorinated solvents. They get energy from it. And there's some that only can dechlorinate, and others, like these organisms, that can actually use other electron acceptors, iron or nitrite or sometimes sulfate. And you can see that these more versatile organisms have big genomes. The restricted ones have relatively smaller genomes, but this is the reductive dehalogenase homologous genes. These are genes that look like those that have been characterized that can do the reductive dechlorination. There are many that are uncharacterized. So presumably these organisms could at one point in time do a variety of different compounds. So just to summarize this quickly, I think that we mustn't forget that the aerobic hydrocarbon bioremediation is actually commonplace and is practiced now. All the big oil companies are doing this. This is how they deal. Anytime you have a tank pull at a gas station, you see a pile of dirt under a tarp, that's bioremediation happening. And so this is a commonplace and probably the most common use of bioremediation, but you do need to have oxygen somehow. So it's done in shallow areas or soils that you can till, and this kind of thing. And then for the chlorinated solvents, this notion that you can do this anaerobically was a big departure at the time. People didn't, the notion of doing something without adding oxygen just seemed like foreign. But we realize now why it works is because these organisms are niche specialists. That's all they can do. So you inject them into the ground, and they're not going to eat something else first, and then that's what they do, and then their numbers will die back down when the chlorinated solvents are no longer there, and that they are common in all environments, these kinds of organisms, but not necessarily those that can dechlorinate vinyl chloride to ethene, which is the big detoxification step. So that's why bioaugmentation might work. And we've been working a lot on benzene as well, because it seems to be a bit of a tough nut to crack as well. And it looks like the organisms that degrade benzene are not the same as the ones that degrade toluene, and they're not the same as the ones that degrade other. And so that again, if something is limited to a few specialized microbes that are just not very abundant naturally, you're not going to see the effect of that activity in an engineering or a human lifetime span. And so we need to, so there may be some room there also for bioaugmentation. Okay, any questions? So I've been back to academia for some time at this point. I was working with Dave and still do, but and all the others they're now at Geosyntec Consultant. At the time they were somewhere else, and I did like consulting, although I didn't like filling out timesheets and things like that. And so it was nice to get back into academia where you could just be a little more free. And at the University of Toronto there was a really strong pulp and paper center. And so I started getting interested in a little bit about what happens at pulp and paper mills. And these are anaerobic digesters. So I didn't do it. I was going to calculate how many methanogens might be in there. But this is maybe 30 meters tall by, anyway, you can see a truck down here. Like there's a person, right? So you can imagine how tall these are. So it was a microbiologist from McGill, David Cameron. I don't know if anybody remembers him, but at any rate, he went and worked at Tembec. This is in Temiskaming, which is at the headwaters of the Ottawa River, just 5 hours north of Toronto. And they have this old pulp mill that's actually a wonderful model of a biorefinery, because a pulp mill is essentially what we're dreaming about in this cellulolytic biorefinery idea. They take wood and they turn it into paper, but in the process they solubilize a lot of the sugars from wood, and then they have to deal with that. And for a long time, the Tembec mill, this particular mill, fermented the sugars in their wash water from their pulping operations and made ethanol and sold it for kosher vinegar production. They also make lignosulfonates, because they have a sulfite mill at the place, and the lignosulfonates are sold as binder. And they also have condensate waste. So they have big condensers and part of their recovery boilers and things like that. And the condensers are full of methanol and acetate, which is just rocket fuel for methanogens. And so these big, huge digesters were anaerobic digesters were installed, I'm going to say maybe a decade ago, but it was because of this PhD microbiologist that went and loved nature and fishing and went out to Temiskaming and started working for Tembec and started realizing that they had this one waste line that was this rocket fuel for methanogens. And I remember getting samples of that and doing it in the lab, and it's just acetate and methanol. And it just methane, right? And so they built these huge PAC reactors as a result of this kind of work. And they're really actually quite complex inside. There's a type of bioreactor called internal circulation. There's a 3-phase separator at the top to separate the gas bubbles from the solids, from the liquid, and all this kind of stuff. And they're really quite involved. But and when they first set them up, there were hiccups. There's no question about it, because how do you start up a reactor this big? Where do you start? Where do you get the inoculum from? You can't, and the mill is producing wastewater at a rate that never goes away. And if you don't have this working, where are you putting it? So the operational challenges are phenomenal. But they stuck with it, and now that it's working really well, and in fact, the granules that you get out of here are just incredible, because they started off just putting this effluent from the evaporators, but over the years they started putting other effluents that they have that might be a little more toxic, and have enriched the microbial community within here to handle more and more complex things. It takes a long time, but you can certainly acclimate the microbial population to more and more things that would be toxic to a different system. This tank here in front is actually an equalization tank. So one of the things you have to worry about in anaerobic processes is have a flow rate or a flow composition that's relatively steady going in. And one of the challenges with the pulp mill is that 1 week they're pulping softwood, 1 week they're pulping hardwood, 1 week they're doing this or that. And so the composition changes dramatically, and you have to be able to manage that. And then not a few years ago, 4 years ago, I met this person. So yes, Nigel is a PhD student. He came to me after 40 years in the solid waste management industry and said, I'd like to do a PhD. And he had an idea. And he, so much organic in Canada anyway, a lot of organic material goes to waste, goes to landfill. And the landfill gas capture efficiency is dismal, maybe 10 or 15% or something of all the methane, and the rest of it is just released. And so it really does release a lot of greenhouse gas equivalents. And so it really is, a landfill is basically an anaerobic digester, but it's just a really bad one. If you try to, the way landfills are moving now is to have less and less organic and not put any water, so then essentially nothing will happen in them. They're going to just be tombs rather than anything that's regenerating or getting better. So he had this idea from his experience to build something called a sequentially fed leach bed reactor. And he had this diagram, and he actually had he did an MN project before this that took him 12 years or something while he was working full time, and had made this design. And the idea behind it is that, that solid waste, food waste, and the green bin. Think of your green bin waste, but with a lot of paper mixed in and all that kind of stuff. This sort of average municipal organic waste. It's really hard to mix, and it's really energetic. It costs a lot of money to mix and a lot of money to grind and this kind of thing. So let's just not do that. Let's not worry about sorting or mixing or anything. Let's just stick it in a thing a bit like a landfill. And then instead, we'll recirculate the leachate. So what he has here are these leachate tanks, and underneath the leachate goes back. So there's a sort of a header that goes around, and twice an hour, leachate percolates through each of these. Okay? And then moreover, he feeds them sequentially. So this is fed on week 1, or sorry, maybe down here. This one's fed week 1, this one's fed on week 2, this one's fed on week 3, 4, 5, 6, and then when this one's 6-week-old, it's taken out and the new one is put in. So there's a 6-week cycle. And then we also take the leachate through a UASB, which is an upflow anaerobic sludge blanket reactor, to sort of maintain or to further digest any of the liquid organic that's coming out of the leachate, of these leach beds. So the actual picture looks like this. This is set up in a walk-in fume hood in the lab. And these are about like that, 15 liters, I don't know what they are. And they're filled with solids, and then the header for the gas. There's another picture. So anyway, this is the UASB here. These are the leach beds. Ignore this leach bed, that's just a spare. These are just peristaltic pumps, and these are the tanks at the bottom. And so the leachate goes down through the UASB into this tank, around, and then there's these little automatic valves here, and twice an hour, 600 mils is spilled into each one. This is showing the piping diagram, and there's these 2 gas meters that are just these wet tip meters that collect. And the really neat thing about this is that it's working incredibly well. So he's been running this thing for 70 weeks, okay? And it's been he's been trying different amounts of food waste to fiber, because fiber is like the cellulolytic kind of things in the food waste. And the food waste, he gets it from Miller. He used to work at Miller Waste. He's just getting the raw stuff, with chicken bones and everything else, and sticking it into these reactors and testing different. And what we definitely see is something he's calling synergy, but basically that the individual wastes alone produce a certain amount of gas. When you put them together, you get more than the sum of the individual wastes. So there is a synergy that happens when you mix them together. And we're exploring that. And here's Nigel. This is what it looks like at the end of digestion. And he makes a nice mixture. And so really the idea is low, reduced capital and operating costs, no mixing. The city of Toronto just has a green bin program, and they just installed these huge anaerobic digesters for this near the airport. But there's no way they make money. They have these centrifuges to separate things. They have these big grinders to grind everything up first, and they put a lot of water in it, and then it just turns into a wastewater treatment plant. Because that's what they create. So I think this has got, if you have the, oh, I went too far. If you have the size, if you have the space, this is definitely a much better option. Okay, I'm getting towards the end. I just find myself going around this circle over and over again now, it seems. So recently we started working on fuel ethanol plants. You know, in Southern Ontario, as you know, there's a number of these. First generation, using corn. It's exactly like making whiskey. It's making whiskey all over again, except instead of making stuff you can drink, you stick it through a molecular sieve and mix it with gasoline, so it's denatured, and stick it in your tank. And there's another neat thing, though, that I learned about fuel ethanol as opposed to beverage ethanol. There's some changes in the cooking process. Okay, they actually use some because they can cook more aggressively and not worried about flavors and the taste of the gasoline. But the other interesting thing is, see, this thin stillage here, they now send it to an anaerobic digester, and that anaerobic digester is then the whole effluent from that is then recycled to the front and back in. And so they use anaerobic digestion within these bioethanol first-generation plants as a wastewater treatment system that actually closes the loop. Because before that, they would recycle a little bit. You can recycle some of this thin stillage, but it gets high. It's high in acetate. It's high in lactic acid. It's high in other things that come from the fermentation. And you treat it with digestion, and then you can send it back, and it doesn't affect your fermentation. Because whatever you send back, you got to worry about what it's going to do to your yeast over here. And so this was a project we were involved in. And so back to Perry, and to just talk about, he's still active, and he's still working a great deal in anaerobic digestion. And this is some old notes of his. He uses funny notation, these equivalents. But the bottom line is that one of the huge advantages between an aerobic and an anaerobic process, when it's using the same substrates, is the yield of cells, okay, is so much lower in the anaerobic system. So without oxygen, less energy is available, but fewer cells are synthesized, and it means less sludge to dispose of. And this business of sludge disposal is becoming critical, I would say, in many environments where we have huge cities and huge amount of wastewater, ginormous wastewater treatment systems. And so a few years ago, Perry published this paper. I think he was 80 years old at the time, but he's still first author on a paper. Think of that. And this was from some time he'd spent in Korea as a visiting professor. And the idea is to replace secondary treatment, aerobic secondary treatment, with completely anaerobic secondary treatment. So to take your wastewater coming in, maybe do primary settling just to get rid of the big stuff, send it directly to an anaerobic digester. And you can do that if the wastewater is strong enough, meaning has high enough organic in it. And that's certainly true in big cities, and especially in emerging economies and so on. And then you have a challenge of you have to get the methane. A lot of it, there's such a high volume of water that a lot of the methane will be dissolved. So you have to strip the methane to be able to use it, but you can still do that. And then there is a challenge, too, that the wastewater will be still high in nutrients. So you do need to either treat the wastewater for removal of nutrients or use it in agricultural settings directly. And so this idea of anaerobic secondary treatment for wastewater is really taking off. And in fact, in Brazil, where I've been a few times, there are so many cities that don't have any wastewater treatment at all. And they're just starting now. First thing they're building is a secondary anaerobic digester for their wastewater, because it just makes more sense. You can, in this paper, Perry calculates that you can actually have wastewater as a net energy producer. as opposed to costing the municipality. And so I think this is the way, already in Brazil there are some 800 anaerobic digesters set up on municipal systems. And so I do think it's something that is needed, and that the old way we do stuff is so energy intensive, and it was built at a time, it certainly achieved the purpose, but I think we could do it for less energy now. And so, in conclusion then, we need modern, ancient biotechnology as well, is what I'm saying. And so we need to bring our modern tools to better understand these processes that are very robust because they rely on these microbial communities that have these synergistic effects, synergistic and syntrophic effects. We need processes that are efficient, so we have to look to what nature already does and tweak those rather than reinventing the wheel. Nature has had more time to work on this. And we need to really think about the volume and the size. This is a big problem in the biofuels side of things as well, which is the amount of material you need to make to offset or to make a dent is huge. And similarly on the wastewater treatment side of things, you can't sterilize, your feed. So if you want to make, products from CO2 fixation with algae, well, you got to be able to grow that algae in non-sterile feed, right? Not the wastewater and the flue gases and things like that. So, and the last point is something that we need to really mitigate within the community at large and the people at large, is the hype then promises that can later undermine a good technology. So I saw this happen in the bioremediation in the 80s when, oh yeah, we're going to be able to design microorganisms that degrade everything, not a problem, no, and the papers in bioremediation went zoom through the sky, and then nothing was working, and so that it kind of fizzled. And yet there are, when the idea of using organisms like the Dihelicocoides came into place, we had to fight against that preconceived notion that it didn't work, okay? And it was because it was oversold in the first place. And I think we tend to do this a lot, and more. So we have to be careful. And so, as I said, just in conclusion then, this anaerobic secondary treatment, I think is coming around again. And I'm going around as well. I'm working now with serum. It turns out that we thought we had fixed all the problems with gasoline decades ago, but actually, no, there's still lots of gasoline plumes around. And so we're working again with serum. We got some funding through Genome Canada to look at this anaerobic benzene transformation in the field. And we're doing that with some gasoline end users, gas station end users, and Federated Co-op in particular out west. So I'd like to thank lots and lots of people for everything here, and take some time to recognize that this has always been partnerships with all over the place, particularly with industry. It's always good to see what the problems are in industry and try and find a way, the sweet spot that intersects with what you can do in the lab or work directly in the industry. And then the funding follows. I'd also like to take a moment to tribute to my PhD supervisor, Dunya Gabor-Galic, who unfortunately passed away months after I defended. She had scleroderma, and it took her at the prime of her life. But she started, she was from Zagreb, and she was an anaerobic microbiologist trained in very classical European microbiology, and was working on benzene and lignin biotransformation in the early 80s. And now everybody's doing that again. and she was one of the most kind people you would ever meet. So I just wanted to, and unfortunately, you won't see that many papers by her because she just wasn't around long enough. So, but certainly helped set me on the way, and was a microbiologist in this engineering department. So.
Thank you very much. We have time for a couple of questions, and then is there a reception after? Yes, yeah. And then we have a reception. Oh, I'll actually be in here, so yes.
Do you have a chance to look at advanced oxidation treatments?
Have I looked at them a little bit? Not yet.
Because they play an important role in the degradation of solid waste.
Yes. And so the idea, I think, is maybe to use combinations of treatments. So advanced oxidation treatments which particular one what are you thinking of in particular?
UV.
UV?
H2O2 and ozone.
And so again, it's a cost analysis and mixing problem as well comes in a lot, but it works for sure.
Yeah, I know it's costly, so.
It is costly, and so we have looked a little bit. I haven't done the advanced oxidation part or the ozone part, but we're looking at whether pre-treatment with ozone or any kind of oxidant, and then subsequently looking at whether that makes things more digestible. The data are very mixed on that. It's not necessarily true. It doesn't actually necessarily help, and it's not entirely clear why. One thought is in these advanced oxidation processes, you perform radicals, and then rather than making smaller molecules, you might actually polymerize some of these molecules and actually make them bigger, which may make them more recalcitrant, which might be okay too, as long as they're not degrading. Yeah.
Sorry, and we are recording for our YouTube channel, so that's why I got the microphone. So I can catch all your questions. Anybody else?
I saw your equation, you have benzene and water.
Thank
In some of your equation, you have benzene and water,
Yeah.
but they are not miscible, right? So how do you...
Oh, that's just the stoichiometry of the equation. But benzene is soluble in water. It has a solubility of about 2,000 milligrams per liter, and so that's actually quite soluble. It's not completely soluble, but it is soluble, and that's why it's a problem in groundwater. It gets into your water, and the drinking water limit is maybe 5 micrograms per liter, and yet it dissolves at 2,000 milligrams per liter. And so, yeah, you don't want to, it's enough for microorganisms to grow on. And then if you have 2 phases, so that's, I didn't explain that well in those pictures. This jargon that we use in the groundwater world is LNAPL, or light non-aqueous phase liquid. And so you have a 2-phase flow, like your oil and water mix. You have 2 phases, oil and water, but as soon as you degrade some in the water, more will dissolve, right? And so the concentration at equilibrium will always be the same. Yep. Yep.
First of all, it's on. Welcome back to Anaerobic Digestion and Fermentation. I was surprised to see you call leachate bad. System.
Can I put it back on?
Yeah,
This?
yeah, I love it.
Okay.
I can see the true contribution economically, like
Yes.
cost saving, right?
Yes.
So, did you ever try this system for other wastewater stream, not food waste?
No, we haven't tried it for anything else, but no. And this is not wastewater, right? This is solid. This is sort of, in Europe, there's already a lot of solid digestion that's happening, or that they take solids and mix it in water and then digest. And I mean, obviously, food waste is digestible. There's no question about that, to some extent anyway. But the difference, so what Nigel, the student who's doing this, he creates a blend of fibers, boxboard, cardboard, paper, and then also food waste. And he just puts them in known amounts of COD into each reactor, and then operates it as part of this, changing them. And so he's slowly, on a 6-week cycle or so, he'll change the composition of how much COD is in these as a percentage of the total as food waste, for example. And that's what he's doing for now. So we haven't tried different, so the food waste is already very mixed. And the idea is to, the idea is to mimic or to copy what a typical municipal solid, organic solid waste fraction would look like, right? That's what he's doing. But so when you said liquids, I'm not sure what you.
Like, I just imagine like thin stillage.
Thin stillage, yeah.
Probably good candidate.
Thin stillage would be good, although it's very wet, right? So this is really amenable to things that are maybe 80% or something like something that looks like I don't know what, like your green bin waste, or maybe a bit wetter, like very thick oatmeal or something. Because it has to sit in there, right? So At the bottom there is a geotextile membrane. It has a sit in there. It has to be held. And he puts wood chips in there to have enough permeability for things to go through. So, but it's very low tech, and yet there are some really interesting things. One of the things we've been doing actually are enzyme assays, believe it or not, on this, because we're trying to identify where this extra gas is coming from, what is actually being degraded. And we find that when you have enough food waste in the system, you really do. And we think that there are esterases and proteases and enzymes that are coming from the food waste that are actually enhancing the degradation of other components that are in there. And even the glue that's in box board, like your cereal board and stuff like that. And so he's also done a whole bunch of those, those BMP tests that I showed you that picture of. He's done a whole bunch of all the individual ingredients in BMP tests, in biochemical methane potential tests, in those serum bottles to understand the contribution from each individual substrate that he then puts in there. And so even though this looks like dirt in a bucket, which it is, like garbage in a bucket, right? Food waste in a bucket, when you start doing the mass balance and trying to understand where is that gas coming from, in a complex situation like that, it's tricky. We've had a lot of fun. Yep.
So the project in Korea.
Yeah, okay.
Okay. I find this very fascinating from making a whiskey now making the fuel ethanol. Would you like to share a little more about this new direction you're interested in the research?
On the fuel?
Yeah.
Oh, so this isn't something, this part here? This one?
Yeah.
So this is actually what happens. There is a Lars Angenent at Cornell. I don't know if you know him, but he's been done a bit of work on thermophilic anaerobic digestion of thin stillage. But if you just, in the industry, even all of the first generation biofuels plants that are in North America virtually do this now, I think. And it's just a way of dealing with your thin stillage. And you just treat it anaerobically, and you get some methane that helps offset your costs. But it also really, most importantly, means you can recycle more water, because a big cost is water, and you have to basically solubilize your flour back. And so they've always recycled water a little bit through centrifugation and just taking the supernatant and recycling the water. But that water had too much organics in it, and so this is just making that a whole lot better. So it's not a new direction. It's not we were just helping them understand their anaerobic diet. They were just so frightened of this thing, and how it works and what might it be doing to their system, like what might it be contributing to the system, that they wanted us to have a look at the microbial populations within.
Thanks.
So I was just asking about the project in Korea.
Oh, yeah?
Did they replace the aeration tanks by anaerobic digestion? Is that
so there
the main
is
thing?
a plant. There is a pilot scale plant. So Jae-ho Bae, the second author on there, is a professor at Ina University in Korea, and he was one of Perry McCarty's PhD students. And he's told me to come and see it. I haven't gone. I don't know if anybody's been, but they have a pilot scale reactor that does primary wastewater treatment. Secondary treatment, like replacing the aeration tanks, exactly. So they have a pilot project that's doing that currently. And also at Stanford, back at Stanford, my colleague Craig Criddle has gotten funding recently for something they call the Codiga Center. So you could look it up online. And I think they're doing the same thing with the Stanford wastewater treatment. They've got the Stanford campus wastewater treatment operators on board. So they've actually taken the sewer line from Stanford, and they've built a smaller version of this anaerobic digester for wastewater, and also separation for the methane. And they're testing it right on campus and seeing if it's working. So that's happening for sure. But in Brazil, I know that the municipalities are setting these up directly as well, and they're full scale. So, and I think actually there's a number in North America as well, a few cities that have set so they're replacing.
Does it require more space when they move?
No, it doesn't. It
anaerobic?
requires less space. Like if you look at this, you can make it as one big tower, right? You don't need the clarifier because you don't get as much biomass. So if you go back to that crazy picture I had, wherever it was. Oh, where was it? Oh, earlier. Anyway, you want all the reactors? You're going to see all it backwards. This one. Right, look at the space. It's something like that. I mean, yes, that's for treating a lot of people, but you know, even Toronto's sewage treatment plant is huge. So no, it can take up less space because you don't need the mass transfer. You need a little bit more residence time. But if you build up your biomass with a membrane bioreactor as opposed to you don't use clarifiers, you use a membrane to do your solid liquid separation. So. There's definitely units full scale that are working. The challenges that you get, you still probably need a little bit of polishing at the end because it doesn't reduce your concentrations to quite as low, perhaps. So it really depends on what the end use of the water is afterwards. And also you might have to deal with nitrogen. And so that's where Anamox processes, I know Josh is working a little bit on that, which is anaerobic ammonium oxidation is also works very well together with secondary anaerobic digestion. So these kinds of processes need to be engineered. They work in, you know, again, they work in the laboratory. You need to engineer the scale and the reliability and the robustness into them. So, but they are being used, and they will be. And I'm sure, like, there's no question that I was at the last anaerobic digestion conference, World Anaerobic Digestion Conference, and that's all I could talk about. Everybody's doing this, so at full scale.
Hi, I was quite interested when you mentioned about the use of bacteria to dechlorinate the water. So what are some of the risks or the negative consequences in using this method?
So it's not dechlorinating the water, right? It's removing organochlorine compounds. So removing a chlorine atom from a molecule that has chlorine on it. So it's not the same thing as the chlorine in your swimming pool, okay? And so the negative consequences, these are organisms that exist in the ground already. There's so many naturally occurring chlorinated or halogenated organic compounds. Even chloroform is produced in vast quantities by fungi and in soils. Antibiotics, naturally, you know, chloramphenicol, it's a chlorinated organic. So these microorganisms dechlorinate organic molecules. That's what they do for a living. And they live there. And so I don't know, we add a few more in where we have a spill of a chlorinated organic solvent that's been made industrially. That's what trichloroethene is produced, you know, chemically synthesized. And so you end up having way more in the environment that the environment is ever used to having. And so to counter that, we put more organisms in so that it degrades faster. And the organisms enriched naturally at these sites as well. So you don't necessarily have to add in organisms. The population that's there, if it's competent at dechlorinating TCE, will grow. So, but they do need an electron donor. They do need, you know, the chlorinated compound, the organic is the electron acceptor. It's like the oxygen. But they need something to eat. They need a food. So often in the environment, the subsurface is quite oligotrophic. There's not a lot of organic matter. And so the organisms are just at a low number because of that. And then if you add in a little bit of electron donor, or you add in a bit of ethanol or molasses or something, then the organisms will grow. Thank you. Okay.
So maybe we just take one more.
Sure.
I've got one, and actually,
I just
there's
want
still
to
some,
reference, yeah.
there's one over here.
Thank you for the talk. How close or how far do you think we are to have a biorefinery technology or concept to be set as a real, commercial technology based on lignocellulose feedstock?
We have, the technology already exists in it, and there were many full-scale plants ready to be built. It's an economic issue at this point in time. It's about how much are we willing to pay to do that. And are the economic incentives there? And so, if we had a price on carbon, maybe it would be economically viable. But it just is trying to compete against natural gas that's free, virtually, at this point in time. That it's really tough. Actually, it's interesting though. Even though natural gas is so cheap these days, the interest in anaerobic digestion and these kinds of things still is very high in industry because also of the solids reduction side of things, and solids handling is a huge, huge problem as waste treatment, municipal ones like these ones here. Like imagine this aeration basin here, think of how much biomass you're making in that, and then you have to deal with that sludgy, gunky stuff, right? And so, and the same goes for industrial wastewater treatment. So there is a lot of interest driven by things that you might not think are. So it's not so the problem with biofuels is that the product is so cheap. It has to compete on a market that's so cheap. Beverage ethanol you can sell a lot higher, so we should all be, making better. So that's why the biorefinery models and the places that do have set up full scale are ones in personal care products. They're making perfumes and scents and things that they can sell, or they're making things they can sell at a high, nutraceuticals that people will buy. They're things that you can sell at a high value because the economics. But it's not a technical problem anymore.
Okay, thank you.
One more.
2 years ago, we had a company here called Enerchem, and they were doing a project in Edmonton on municipal waste treatment, which seemed sort of fantastic. Do you know how well that's working, or?
Yeah, I think it's working well. It's a combustion technology, right? Isn't it? And they're making biogas from waste, from garbage, and I think, and it's. I know. I'm trying to think of the researcher's name who started Enerchem. I've met him. I know. He's
father and son.
It's a father and son, exactly. Anyway, this is going to be recorded, and then he's going to find out. So you're going to have to blip this out. But it's a very good technology, actually, and again, it's about the bio oil and the gas that you make. But certainly, think of the waste you put in your green bin. There is so much carbon in there, and there's so much locked up energy in all of that chemical. And that there's got to be a way to get it into something that we can use. And that's just another way. And so essentially, microbial reactions are a very slow combustion. And I think that the Enerchem process is a pyrolysis. I can't remember exactly. I think it's a pyrolysis process, and it works really well. But again, it has to live in this climate of very cheap oil and very cheap natural gas and energy, and it has to wait for that to shift a little bit, I think, before it'll be really take off, or some regulations to change for that to take off. Charnay, that's it. Yeah.
More?
Was there one over there?
Yeah.
So I just had a question about the solid waste treatment experiment you're doing, because my understanding was that's on a 6-week cycle for each bin.
Yeah.
So how would that scale up to a municipal level?
So the idea, they're actually, I think, building, or they're trying to get permitting to build something on a pilot scale in Ottawa, because I think the idea would be you'd do it on a landfill, a bit like a landfill. So you would have cells, and lined cells in a landfill, and you would dump the day's green bin waste, or not green bin, not green. It is green bin waste. So the food waste in there that's been collected, but actually it's more what's been collected. It contains paper, and anyway, put it in there, and at the end of the day, or at the end of when that cell is filled, it would get sealed progressively, and then you'd have a membrane over it, and then you'd have a leachate collection, and you'd recycle it, and then you'd fill the next day, next over. So the cycle might not be 6 weeks. It might be a little bit longer in the context of real waste that's going to have more heterogeneity and larger larger sizes and things like that. Because here we do remove the big stuff that won't fit, but that's the idea. It would be operated a bit like a landfill, but that would be managed, and the thing that's offers the synergy primarily is that you have fresh fresh waste. The leachate from that is very rich in microorganisms and these enzymes that I'm talking about, and that's recycled onto a part of that would be very old, and that where most of the readily degradable stuff is already gone, and you're just left with a lot of the cellulitic material and I don't know what, gristle, and I don't know what else. And it seems like you can get an extra boost of energy or gas out of that if you're recycling leachate from a fresher reactor. So that's what it's offering. And we think that's definitely true. So it'd be worthwhile building that plumbing into a landfill. So it'd be basically a landfill digester model. And so it could be quite low cost. Maybe you'd put some cement burns or not, but maybe not. Maybe you can just do it with geotextile membranes. So, yeah. I could put you in touch with Nigel. He would talk forever, yes, about it. But we would love to try it in a larger scale.
Great. So I think that we should move on to the next part, which is going to be a reception in this room. I'd like to thank Elizabeth again for really a talk that gave people, I think, a lot of ideas. And it was nice that you presented it in the context of your career and how really everything is connected. That's something really for the young people in the audience to think about and to realize. We look forward to following you in the years to come.
Oh, thank you.